The Elisa assay exploits the antigen-antibody affinity to signal the presence of a molecule of interest. In this case, ELISA is being used to measure the inhibition of HIV-1 capsid dimerization. The HIV capsid forms when individual capsid proteins dimerize at the C-terminal domain. This lab had previously shown that the isolated C-terminal domains, or CTD, will still dimerize since they retain the residues directly involved with binding, but that the binding can be disrupted by another CTD molecule. This assay will measure the binding between two CTD proteins tagged with either GST or flag in the presence or absence of untagged CTD. A color-based enzyme will provide visible conformation in proportion to the intact dimer that results, and measuring the absorbance will allow us to quantify the level of dimerization in each condition. The assay is performed in a 96-well plate to minimize reagent waste and provide an optical surface that allows accurate absorbance measurements. First, the plate must be coated with the first half of the dimer, in this case GST-CTD. Some wells are coated with plain GST or PBS as controls. To ensure full and complete adsorbance, the plate is covered and left to incubate overnight at 4 degrees Celsius. The next day, the plate was washed 4 times with a solution of PBS with 0.05% tween 20. This removes any non-adhered proteins and prevents interference. It's important to firmly tamp the plate on paper towels to remove as much of the wash buffer as possible. Next, the wells are filled with a blocking buffer of PBS with 5% milk and placed on a rocker to incubate for 1 hour at room temperature. The milk contains proteins that block nonspecific binding to the well itself, preventing excessive background absorbance while stabilizing the absorbed protein. After blocking, the plate is once again washed 4 times with PBS tween to remove unbound milk proteins. During the blocking buffer incubation, the 2 CTD proteins and a standard CTD control are diluted to 0.5x, 2.5x, 5x, 10x, and 50x in PBS milk. If the proteins behave as expected, the absorbance readings should increase proportionally with the CTD concentration. Before loading, CTD flag is added to each sample to provide the other half of the dimer and bind to the GST-CTD. The proteins are loaded onto the plate. Several controls are put in place to verify the proper functioning of the assay. CTD flag is added directly to GST-CTD coded wells to verify complete coding, and also to a PBS coded well to act as a negative control. Separate 50x CTD dilutions are prepared without CTD flag and loaded into PBS coded wells to signal if nonspecific binding occurs to the well. And CTD flag is loaded into GST coded wells to signal if nonspecific binding occurs to the plain GST. After loading, the plate is returned to the rocker to incubate for an hour at room temperature. 4 more PBS tween washes remove unbound proteins, and the plate is loaded with the primary antibiotic. Most wells receive rabbit anti-flag, which will bind to any bound CTD flag, but certain wells receive rabbit anti-GST because no flag is present. The plate is returned to the rocker for another 1 hour incubation at room temperature. Another 4 PBS tween washes remove any unbound antibody, and the second antibody is added to bind to the primary antibody. In this case, it's goat anti-rabbit, which will bind to both rabbit anti-flag and rabbit anti-GST. The secondary antibody is tagged with horseradish peroxidase, or HRP. HRP is an enzyme that produces a blue color in the presence of its substrate tetramethylbenzidine, or TMB. This color change allows us to quantify the level of binding by measuring the absorbance of each well. After another hour on the rocker at room temperature, and another 4 washes with PBS tween, an additional wash with plain PBS removes traces of tween 20 that could cause background interference with HRP detection. Next, the TMB is added to all wells to bind to the HRP. The plate goes back on the rocker, but this incubation is only 15 minutes long. Finally, stop solution is added to all wells to prevent the HRP-TMB reaction from proceeding any further. The plate is loaded into the microplate reader, and the absorbance is read at 450 nanometers. The data from the microplate reader is loaded onto a spreadsheet. The triplicates are averaged, and the averages are adjusted for background absorbance to provide a true absorbance value for each well. This assay shows that the control CTD prevented dimerization in proportion to its concentration, although the lowest concentration shows a lack of consistency signified by large error bars. The 2 CTD samples being tested show low absorbance, which corresponds with low binding, which means high inhibition, but the consistency across concentrations is an unexpected result. Rather than take these results at face value, either the test should be repeated or a different assay should be used.